GM1 gangliosidosis is an ultra-rare disorder caused by a mutation of the GLB1 gene, resulting in a deficiency of B-galactosidase, more commonly known as lactase. Lactase catalyzes the hydrolysis of GM1 gangliosides into monosaccharides through the breaking of glycosidic bonds, and thus a lactase deficiency leads to accumulation of GM1 gangliosides. This accumulation occurs in organs throughout the body and progressively destroys neurons, particularly in the brain and spinal cord. The effect is progressive degeneration of the central nervous system with cognitive impairment, paralysis, and early death. GM1 disease is inherited in an autosomal recessive pattern, as seen in cystic fibrosis and sickle cell disease, which means that affected individuals have received one defective copy of the GLB1 gene from each of their parents. It is estimated that GM1 disease occurs once in every 100 to 200,000 newborns. However, it may present at any stage in life. Based on the age GM1 disease presents, it is classified into three forms: Type 1, infantile; Type 2, late infantile and juvenile; and Type 3, adult. There is currently no approved treatment or cure for any form, and subsequently research into finding an effective therapy is imperative. Gene therapy is not a new avenue for treatment, with the first proposal for its use over 50 years ago and the first successful trial over 30 years ago. However, recent work has revealed that in feline GM1 models, injection of functional brain tissue with an adeno-associated viral vector, or AAV, provides noticeable therapeutic benefits. This knowledge sparked the research of Gross in 2022 in the study Intravenous Delivery of Adeno-Associated Viral Gene Therapy in Feline GM1 Gangliosidosis. The study aims to ultimately set the foundation for future approved treatment in humans. The presentation of late infantile and juvenile GM1 in both humans and felines are comparable, so therefore they were selected as an effective model of the disease. The use of feline model is to enable the evaluation of the candidate therapeutics before possibly initializing clinical trials. Gross sought to develop a less invasive treatment for the brain and increase systematic biodistribution through evaluating intravenous injections of AAV9. The experiment used AAV9 vectors with a DNA backbone for expressing B-gal enzymes. The vectors also contained a hybrid chicken B-actin promoter that was included for expression of the gene. The AAV particles were prepared through triple transfection of 293 T cells, which is a cell that is highly transfectable and is derived from the human embryonic kidney 293 cells. Triple transfection is a method of putting three purified DNA sections into a eukaryotic cell, in this case for the purpose of producing a modified AAV. The triple transfection occurs with three plasmids inserted to the 293 T cells. The helper plasmid, FD6, which provides genes for mediation of replication of AAV, the plasmid expressing AAV9 capsid, which is a protein shell of the AAV and will be used to protect genes for treatment of the body. The triple transfection contains a final plasmid that has the feline B-gal gene for expression of the target enzyme. Inside of the 293 T cells, it will begin to replicate AAV9 capsids containing the B-gal gene according to the replication genes of the helper plasmid. Then, using an iodixonol density gradient, which works by having salt solutions of different densities to destabilize ionic interactions between molecules and then separate the molecules based on density, the AAV particles were then extracted from the iodixonol density gradient at their known density and desalted, as to be purified for use. The AAV9 vector was injected into the bloodstream of subjects diagnosed with GM1 gangliosidosis. 5-month short-term and a 40-month long-term group were observed, as well as control subjects with and without GM1. Both long-term and short-term groups were then assessed periodically using a 10-point clinical rating score that was based on a scale of neuromuscular disease severity. Subjects that were determined to have severe symptoms would be humanely euthanized. Otherwise, they were monitored and assessed for 16 weeks post-treatment. MRI and magnetic resonance spectroscopy were used to compare tissues of both control groups to subjects receiving treatment. Genomic DNA was isolated and run in a PCR with primers that isolated the feline B-gal gene from the genome to determine the vector genomes present per cell. The use of viral-based gene therapy to treat neurodegenerative diseases is a well-studied practice, with papers dating back over 2 decades. However, it has been used in multiple other clinical trials to combat various diseases and ailments. The use of viral gene therapy on treating brain tumors has shown promising results in past trials. Two separate studies, Oldfield 1993 and LeMay 1998, published in the book Human Gene Therapy, showed promising results using a different approach to treatment. Initially, retroviral vectors were applied to growing malignant tumors in rats. These vectors contain the RNA of a simplex virus, thymidine kinase, a strain of herpes. The subjects injected with modified RNA are then allowed sufficient time for the vectors to transduce. The subjects were then treated with a drug called gangaclover, or GCV, which is an antiviral drug used to treat herpes. The use of GCV was to target cells that were infected with the modified virus and eliminate them. The study found complete regression of all tumors using GCV therapy, with no evidence of transduction into surrounding brain tissue or any significant toxicity. This presents the possibility of effective treatment of tumors via viral vector-based gene therapy in the future. Viral vector-based gene therapy can also be used to treat physical ailments such as hypertension and diabetic cardiomyopathy. But this is a difficult procedure to execute, as there is a wide array of genes that can contribute to these disorders. A study into treating hypertension showed a significant decrease in blood pressure in spontaneously hyperintensive rats after intravenous injections of AAV vectors were administered. This would be an effective treatment to mediate hypertension. However, the vectors were found to stimulate an inflammatory response. Similarly, viral vector-based treatments have been trialed for diabetic cardiomyopathy, which is a disease that is defined by having an abnormal myocardial structure in people with diabetes mellitus. Intravenous injection of AAVs were implemented to halt the downregulation of the PIM1 protein. The results were significant, showing that cardiotropic viral vectors were able to halt the progression of diabetic cardiomyopathy. The utilization of viral vectors has been shown to combat a variety of diseases. The results from various clinical trials are promising, however there are still some implications and risk present. Somatic gene therapy has been largely given the green light, albeit under strict regulation. From an ethical perspective, somatic gene therapy can be viewed like traditional drugs. Ethical issues include many that are familiar to a clinical setting, such as risk versus benefit, bias versus monetization, and informed consent. The four pillars of medical ethics guiding Western medicine are beneficence, non-maleficence, autonomy, and justice. They can be applied to somatic gene therapy for its use and development. In 2017, a gene therapy for a rare genetic disease that causes blindness became available with treatment to a single eye, costing over $400,000 per year. Many treatments like this, they are unaffordable, which raises the issue of equitability. In the case of the proposed gene therapy for GM1, there is still much research to be conducted and clinical trials to test its viability before it can be considered as a treatment option, further highlighting gene therapy's high cost. However, there are strategies that can be put in place to combat this. Crowdfunding reframes the notion of the burden of cost being placed entirely on the individual. Payments based on certain milestones being reached are already being trialled for some gene therapies available in Germany. Subscription-based models for the access of a treatment to an entire healthcare system also have the potential to decrease the cost of the gene therapies in the future. The use of AAV vectors for gene therapy has been known to cause the addition of nucleotides into the recipient's DNA, causing mutation known as insertional mutagenesis. While it is uncertain whether the proposed gene therapy for GM1 causes insertional mutagenesis, possible techniques to address the issue are being developed. One such technology is the engineering of specific zinc finger proteins and meganucleases. Zinc finger proteins are naturally found associated with the human DNA. Genetically engineered zinc finger proteins bind with a high specificity to certain genes, allowing meganucleases to cut out mutated DNA. A problem with AAV gene therapy is the potential for adverse immune responses in recipients, as in the tragic case of Jesse Gelsinger. In 1999, Gelsinger, only 17 at the time, suffered a fatal immune response due to a high dose of AD5 vector. Similar concerns exist for AAV vectors at high doses. If immune response to GM1 gangliosidosis therapy proves to be a problem, cloaking may be the best way around this. Cloaking is a method that can be used to decrease the immune response to AAV vectors. It is done through incorporating short, non-coding strands of DNA into the AAV vector. This has the effect of decreasing activation of toll, like receptor 9, of which, when activated, produces an innate immune and T cell response. The study Gross 2022 calls more research on AAV9 therapy to treat GM1, which will determine any adverse effects on humans. General safety concerns for gene therapy can be solved through an increase in knowledge, helping us further quantify risk and analyze future methods on a case-by-case basis.